Der letzte Vortrag für heute von Rüdiger und Mirella. Es geht um Biohacking.
Biohacking und das Personal Lab.
Das Personal Lab.
Yep.
Hört sich ja mystisch an. Ja, viel Spaß.
Okay. So, my name is Rüdiger Trojok, and I'm here with Mirella Alistair, who's at the moment busy with some tech issues. So I'm just starting. We are Bioflux, a small developer group, and we do biohacking. And in this brief talk, we want to explain you what we are working on and what you could get out of it eventually. So in 1942 in Karlsruhe, my hometown actually, Konrad Zuse invented the computer. I think he was really the first person to build one, and it kind of looked like this. You're probably familiar with this kind of images. It filled the full room and it didn't do too much. Nowadays, many developers, probably some of them are here, are developing computers that look like this, that fit in our pockets. We are using these computers to process code, to read and write program code, text code, and process information that we can use to run our societies. But there's another type of code, that is the bio language. It's the genetic code. And the genetic code we can also read and write, and there's basically two versions of it. There's a digital and an analog code. It's like two sides of the same coin. Because the biology is analog. Like we can touch it, we are analog stuff, we are physical entities. But our instructions, how to operate, how to run, are digital. They're an abstract sequence, this one here, of genetic information, which are embedded in the DNA molecule, which we can sequence and also synthesize, so read and write. And the sequenced version is kind of digitalized the description of what is written in this code, which we can read and process with a computer, and only with a computer. I mean, we can basically read and write these sequences, but they are impossible to translate because they're so large that we can only do it with a real digital representation. So this is when I mean that when I say that the biology is digital as well, it's the way that we see biology. We can only perceive it as digital if we go into the code. Working in the laboratory today means pipetting. A pipette is a thing like this, and it's a pressure valve that you trigger with your thumb, and you're operating tiny amounts of liquids between 1 microliter and 1 milliliter in range usually. This is what you can process easily and observe visually with your naked eyes. And what you do day in, day out as a biologist, I'm a biologist, is that you use these little pumps here and you do liquid from A to liquid B, and then you check it and you do it again and again and again, and usually it doesn't work. So that's the reality in 21st century biolabs. And I think it's kind of ridiculous today. There has been approaches to automate this, and that's why I started with a SUSE computer, because it reminds me quite much of it. So they are full room installations that are basically trying to do the same thing that I do with my hand, but automated with motors and screws and mechanisms and so forth. And it's kind of like, if you think of these football robots that are, like resembling humans, and they walk around and they try to kick balls. So these robots are comparably efficient to what a human does. So they're extremely costly and they don't work too well either. So only if you have the same procedure that you want to automate and you do it all over again for millions of times, they're kind of okay and economic to use, but not really. Like they're not the smartphone or the personal computer that we are having today and that we're used to. with computer technology. So if you think of SUSE computer, this is the analogy. There is no analogy for the personal computer. So we want to do the personal laboratory. And this is the device we are working on. We've been working on it for almost 2 years now. And it's a little Arduino-controlled microprocessor, which is switching high voltage of about 250 volts and passes it on to this chip here. We call it Biochip, in which we are loading our reagents. And now I want to hand over to Miri, who's going to explain you exactly how it works. You have the adapter?
Mm-hmm. Okay. So this is a personal lab that we're working on, and those that have seen us last year, you can see there is a progress from the prototype we had last year. For those that have not seen us last year, we're going to show it in action, and this is what it does. Now, as you probably noticed, our technology is based on microfluidics, the magic behind understanding the behavior of small fluids. And when I say small, I mean microliters, nanoliters, and since recently picoliters. So years ago, when scientists started to study fluidics at micro level, they realized that those fluids behave differently than fluids at macro level, like fluids that you have in a jar or in a glass. There are two particular properties that makes them behave differently. The surface tension is insanely big in small volumes, and there is a surprising laminar flow inside. And because of that, a new science was born called microfluidics. Now, with our personal lab, we are interested in the digital aspect of microfluidics because we are targeting devices that actually program the microfluidics. How does it work? Well, we start with an array of electrodes. Electrodes can be made in different ways. The ones that you see here are made using traditional PCB etching, printed circuit boards, out of copper. And the secret is actually to have a droplet on top that covers the electrode and a little bit overlaps with the neighboring electrodes. And then when you apply an electrical potential, also called the voltage, a phenomenon called electrowetting on the electric happens, and the droplet moves towards that electrode. And using electrowetting on the electric, or EWOD, you can also merge droplets and then subsequently mix them, or you can also split them. So, sorry?
How do you split? Merge is simple. If you have different plus and minus, it will attract, but how do you split it?
You just apply voltage on the two neighboring electrodes and then it splits. Yeah. We can go into equations later, but I am willing to answer questions and prove it on paper if it's needed. Now, if you can imagine a little bit that those droplets carry blood, reagents, urine, and proteins, a lot of things that are used in biological protocols today, a full biological protocol can happen on such a device. We just need to work on it and design it. So right now we are setting up a lab, but until then we tried a few experiments with over-the-counter liquids like water. Then we tried to see if our insulating solution is good enough, so we worked with conductive ink. Luckily, we didn't see any sparks happening. And lastly, in case our project fails, we want our personal lab to produce something for us, so we wanted to see if we can make coffee. And we moved coffee droplets as well.
Can you mix chocolate or coffee?
We can, yeah. Sure. Yeah. Now, I know it's fascinating to watch these droplets move, so I'm going to show a little bit more of that. What I want to show here is the lab that we're collaborating with that develops a special type of ink that you can print on simple paper. They showed it on newspaper. This is photographic paper, and then you can move droplets on it. The advantages, probably you've heard already of silver ink, printed electronics. The advantages of this new type of ink is that it's much cheaper than silver. And you can see here droplets being mixed. Yeah. Their platform is currently manual. Our platform is quite automatic. It's controlled through an Arduino, through a software. So we try to put the two of them together. I'm just going to show a little bit more droplets and then... Yeah, they have quite a manual platform. So we try to put the two of them together, and we build this cartridge that connects our personal lab to the new printed ink technology. Is there a pointer? No. So here you can see that you can hold in it. There where I pointed, you can hold in the printed circuit, and the wires that are on top connect to our platform, so it could be directly controlled through the computer, instead of having to move manually the droplets by applying on each electrode voltage. So I'm going to talk about the research challenge. We still have a few years to go into sensor integration. So assuming that overnight to solve all our biocompatibility problems and other things, we'll need sensors on these platforms for two reasons. First of all, we need for feedback. We need to know, we need to have a closed loop to know what's happening there. For example, a lot of errors can happen because of volumetric errors and other things like that. And it would be really nice to have software that corrects that and reacts real time to that. A suggestion that we looked into is to build a capacitive sensor that measures the capacitance on each droplet and on each electrode. We need also sensors for biological purposes. We need to detect things. There are these bioprotocols called bioassays, for example, detecting the glucose level in blood, and for that, an optical detector would be appropriate, we think of it, and we're open to any ideas. So if you have any knowledge, or you know anyone that has any knowledge in sensor domain, or you're willing to investigate something regarding that, we are open for...
Very simple.
Yeah?
Just say open computer vision, which is a library.
Yeah.
Like a cheap camera.
Yeah.
Have some lighting, for example, with ultraviolet light, and you can also see fluorescence. And then you do just picture processing, and you see in which area the droplet moves.
Yeah. Thanks. This is, yeah.
Are there some patents of this? Because it looks cool, so maybe some evil company patented it?
Oh, yeah.
Like
Probably everything is patented, yes.
Can we progress it and make the asks?
Okay.
No problem. And of course, I keep mentioning automation and digital, so I'm going to talk a little bit about the software. What does it mean to build a control software for such a platform? And it means, the setup, as we see it at the moment, is that the biologist or the user designs the bioprotocol, then the bioprotocol is compiled through the software, and the results of the compilation are stored in the microcontroller, currently an Arduino, could be another microcontroller, and then the droplets are moving according to those results. And what does it mean to compile? Well, we start having as input a bioprotocol. This is modeled as a graph, and an architecture, which is depicted there as an array of electrodes. And then we need to place mixing modules where droplets are going to mix, dilute, detection modules, and then we need to schedule those operations. So there are two big problems here, the placement problem and scheduling problems that we need to solve. We are currently working on that as well. Of course, we want to introduce some sensors, so we're going to have a sensor feedback, which means that we need to have a compilation that runs online, real time, so we need smart algorithms that are able to run much faster than the offline ones. And for any technical questions, I'm willing to take them at the end.
I will take over again very briefly. And I want to show you some of the ideas what we think this device that we are working on can be used for. Here we go. What's going on? Whoa. Doesn't want to work with me. Okay. Sorry. So we call it the personal lab. We think the main benefit is that it's automated, but as you've seen before, there is already an automation solution. So on top of that, it offers miniaturization, and it will be comparably cheap and available to many, many more people than before. And probably the main feature is standardization, because to date there is no global standard, only local standards for laboratory procedures. So when a lab comes up, like here, Max Planck or whatever, publishes a new finding in Nature and they make a big fuss about it and it's super cool and so on, less than 30% of the results are reproducible. And that is mainly due to massive lack of standards in biology, because you still manually operate the whole thing. So with the personal lab, we will be able to get rid of all of this kind of old school bio tradition and make it into something that is much more like 21st century style. And we want to call it, oops, where is it? Stuck again. What is that? Okay, it's crashed. So we want to make bioapplications with this, like smartphone applications. We call them bioapps, and we want to run bioapps for sampling the environment, analyzing substances that you collect, grow cells, work with DNA, extract DNA, modify DNA, change metabolisms, so you can basically program whole genomes of organisms to make them produce specialty compounds like proteins or small-scale compounds like organic chemistry molecules that you can use for curing people or medication or whatever. And each of them will come in a special biochip. So every biochip will have its custom design for a specific application, and it's interchangeable. So we have the protocols in an online database, which you can download, and then you can get the specific biochip with certain range of features, and you can run the protocol on that biochip. You can program your own protocols, verify them, compile them, and execute them. So there will be a variety of biochips with a variety of options. Probably also certain combinations. Maybe we will build them larger or stacked or whatever in the future. That will allow the users more freedom. Everything is going to be open source, and I think that's a radical change in the life science industry, because they are not really open source. They're hiding everything, and I think this is what will make this technology more successful, because there is companies that patent the stuff. But they usually don't use it for what we would like it to be used for. They just have their specific pipelines or their machine parks, and they just use this electrovetting feature in one component of a big pipeline of procedures that they come up with, and it's extremely expensive, and it's really not the concept that we have in mind. Because we're a small team, we're just 4 people at the moment, collaborating with a few other developers around the world. We want to open source it, and we hope that many of you spread the word and pick it up and contribute their developments, and that we not stay the only ones doing it. So when if we manage to grow an ecosystem around this technology, I think the big companies now will probably also profit from it, because it stays open and they can continue their own research, and I hope they will see the value of this. So what could it be used for? At the moment, we're having the antibiotics crisis, often in the media as well. This here is a picture from the place where I studied in Freiburg. Freiburg University, just a hill outside of the city. And it shows a Streuobstwiese, apple plantation. And usually people in this area are quite romantic, have a romantic view on nature, and they kind of like their landscape and so on. But there's something awfully wrong here. It's antibiotic resistant genes creeping around in the countryside. Because if you want to productively run an apple plantation, you have to spray antibiotics on your plants, on your trees, like openly spray them in the environment. And that's creating an overuse of antibiotics, which get in the environment. And through that, the antibiotic resistant bacteria emerge with the antibiotic resistance on certain genetic elements, which they are very easily changing from one species to the other. So if you dump a lot of antibiotics on the soil, or also in us or in pigs or farming or wherever you have many antibiotics, the bacteria resist. They have evasive strategies, and they tell each other, kind of like a peer-to-peer file sharing system for genetics, how to get around this. And they do this quite effectively. So they render the antibiotics useless within 10 years after they were used the first time, globally. And the problem now is that producing antibiotics and making them, giving them for use to the population globally is quite a costly procedure. Like researching it, pharmaceutical regulation and so on, make it quite a high cost operation. And the pharmaceutical companies just don't want to pay that bills anymore, because they have better drugs to sell, like heart drugs that you swallow every day for 30 years until you die, and other things that pay quite well, like cancer treatments and so forth. So they just completely dropped antibiotics research in the last 30 years. Since '87, they just don't invest in it anymore. And the antibiotics treatments that we're getting now, and I'm sure everyone had antibiotics treatment in their lives, probably too often, are just the last remaining drugs that are still available that have not been overcome with resistant patterns by the bacteria. So we're really, really in a shitty situation now. And 10 years ago, it would have still been possible to act against it, to do something, but they still didn't think it's worth to invest money in the health of the population. And recently, the governments, like 2-3 years ago, the governments, US, German administration and so on, found out that actually it's a problem, and they started to do something. But what the pharmaceutical companies blackmail them with is say, okay, you extend our patents up to 30 years, and from 20 to 30, and you give us billions, else we don't do anything. And I don't think it's really a feasible solution, because the next time they will ask for 40 years patents and more billions and so forth, so it's not really going to change much. So we need to change the way that we deal with it. And I think one way is the phage therapy, which was invented by the Soviets in the First World War. Phages are little small molecular machines, you could say, that are parasiting on bacteria and just eating them, destroying them, proliferating. So it's kind of a virus, but it's not affecting us at all. It's just for bacteria. They're well known since 100 years. They're very safe. They're highly specific, only to very, very specific organisms they can target, so there's no cross-reactions with our body cells or something like that. And they are programmable. And the programmable part is something that I find here very interesting, because their genome is quite small. They have about 20, 30, 40 genes. You can see them schematically down here. It's in sizes that we can very well handle with organic chemistry at the moment, what we can synthesize. And by programming them, it is possible to change just a little bit, just the patch that the bacteria gets resistant to, because it's the same effect. Phage attacks, bacteria gets resistant, phage evolves, and so forth. So it's always going back and forth. But we can program the part of the phage that the bacteria knows that it's resistant against, and just overcome it very easily. And for this, we need decentralized technology, because it needs to be patient-specific. So every time you would go to the doctor, imagine that you get your own drug treatment, specifically made for you. But it works for no one else but for you. So what we need is a portable, small laboratory device that can diagnose you and that can come up with the drug that you need without going through the whole industry thing. And I think this personal laboratory idea has the potential of getting there. So if we get all the bioapps in place for analysis and drug production, which is very feasible, we could come up with an open source drug production at very low cost. And then distribute it globally and get individual therapies for everyone. If you are interested in the subject, generally referred to as biohacking, you can also get a book that I wrote. You can just biohacking, you get it on Amazon. It's published by Francis Verlag, just 2 months ago. Als Gentechnologie für alle, Werkzeuge der Genbastler, Biomaterial, Geräte, Software zur Bearbeitung von Genen und Praxisbeispiele und Zukunft der Gentechnik. And it's also explaining the idea of the personal laboratory. And all sorts of other questions like security issues, reliability issues, legal issues around it, and also more into depth the technology and how it's going to work. Thanks a lot for your interest. And please sign up for courses. We want to offer courses because we thought that we can do the Red Hat model with this technology. So we want to offer services and courses in the future while sharing all the technology that we're developing. Any questions? Can you come, please? Yeah?
You know that there is a DARPA project now on programming biological cells by VHDL, which is more or less microcontroller stuff and compiled from C to VHDL. There are a lot of theoretical projects, P networks, and things like this, all about bringing together computing, exact computing on the one side, and on the other side biological systems. So this would fit perfectly together.
Yeah, but that's actually, I was asking the speaker too before about the military involvement and if we can exclude this with a licensing scheme, or if it's a good idea to exclude this, because I really don't want to see this in the hand of the military, seriously. And
it simply takes the results.
Yeah, but the way that DARPA works, I don't know. Yeah. Well, if they would be ARPA, yeah. If they are DARPA, rather not.
What about using this thing for legal things, like for example, to produce marks against your body cells or something? So maybe you put them on your face and you look like a zombie because they eat your skin or something. Or you have a label you don't like and you program them to attack the cells of the skin of your neighbor.
Yeah, but phages don't attack us. Like seriously, not. There's about 100 times more phage particles than cells in the planet. So in overall biomass, we have the planet is a phage planet and not a living organism planet. So they're all always around. They have always been around. Probably the first living forms were kind of phage particles, actually. So that's really not something of big concern. Like the phage per se is really not a problem. And the antibiotic resistance patterns are stretches of like 20 base pairs that you would change in an existing phage to overcome the resistance of the bacteria, but only of this bacteria. There's really no chance of cross-reactions there.
Companies who make biogas and you produce plagues against the bacteria in a biogas plant.
Yeah, what about Kim Jong-un in North Korea? I don't know, sorry. Like these questions are a bit far off. And since we want to do everything open source, it's way too complex to just do it in your backyard. I mean, we are just making a very first incremental step, and we are relying on other people to contribute and take it another incremental step further. And if it's so open source, you will spot easily if someone is going down the wrong train. And it's not that stuff like that happens overnight. So the security thing is something that you can read about in my book as well. I was advising the German parliament on the subject as well. Yeah, but the fact is that it's very complex and you cannot just hide it away. So if you're going down the wrong train, you will be probably spotted early in the process. That's what I'm saying. It's open source and it's too complex to do it in your backyard. You will not progress. It's really, really complex. Yeah.
How much time do you put into this? Are you doing this as volunteer work? Are you using these courses to be able to do this full time?
Well, at the moment it's volunteering work, and we would like to run courses to get it a bit more off the ground. And we also plan to develop, to come up with developer kits that people can buy it from us and so on.
No, there's one more. Okay,
One, two. Anyone else? Okay, you there? Second. Yeah.
How's with financial support? So I think you need some.
Yeah, if you have money, you're welcome. Yeah, we.
There's some institutions which are interested in these techniques.
We are looking into funding opportunities. Yeah, so we're trying to get more funding from the government and things that are not so binding in terms of what is after product, because not all of these institutions that you mentioned are okay with open source or releasing things after that open source. But yes, we are looking into funding opportunities.
You use the technology. Is there any continuation of the last sensors? Because the drop not always moves with all their.
The risk of contamination always exists. There are two ways to go with it. Either you can control from the software where the droplet is located, and then if you have two operations with contamination constraints, you constrain them to several areas and you make sure that they don't intersect. Or you schedule a wash droplet that goes and mops around. It's like a mop and just goes and washes the area after the droplet. These are the two solutions we have found so far. We are still looking for that. But yeah, contamination is definitely an issue. And
it's going to be not too expensive, so after X amount of repetitions, you just get a new one.
Thank you.
Yeah, sorry that I have to close down the session. So, Mario, one of our main